And you guys ready to get started?
Yeah. Okay.
Cool. Right. Welcome to session 31 of BGA24. This session's going to be on gfastar with Giulio Formenti and Jack Medico. Please take it away.
Thank you, Damon. Thank you so much, everyone, to decide to join this session. Hope we can make it interesting for you and a bit interactive. So the way it's going to work, or the way we thought about this, is that we're giving you some background on these tools that we're going to present, and then we switch over to GitHub, sorry, to Gitpod to uh, try them live. Okay, so I'll try to share my screen now. And here we go. Let me just hide some of all the controls here. Okay, so both Jack and I are from the Vertebrate Genome Laboratory at the Rockefeller University in New York. And our focus is really on genome assembly, generating reference genomes at scale. So we started developing our own tools internally for basically assisting this process. I don't know how many of you attended other sessions. I don't know if in these 31 previous sessions, there was a mention of T2T as a concept. But we'll talk about it briefly today as well. So the tool suite is called gfastar. GFA is the keyword here because GFA, you may have heard or you may already know, it's a file format to store assembly graphs. And star is just to say we can try to do a lot of different things. I just want to mention, because it's news that makes me happy, and also I think will suggest that there will be more and more support in the near future for this tool suite. So we were able to secure a small research grant from NIH. It's our three, with two aims. One is to continue to develop basically a general purpose library for genome assembly, evaluation, and manipulation, and then develop specific methods embedded into tools for genome assembly at scale. So this library and this tool suite is designed in, sorry, I'm skipping slides, but I shouldn't do that. It's designed in C++, and I'm sure some of you are already familiar with that, but I'll come back in a second. Just to say who we are again, so we're part of this international network called the Vertebrate Genomes Project. It's part of the Earth Biogenome Project. We have been organizing the BG conference for a number of years. Our goal is to generate reference genomes for all 70,000 vertebrate species. The Vertebrate Genome Lab is one of the main hubs generating the data. The Sanger Institute, where Damon is another one. And then there used to be the MPI, is no longer generating actively data, but has been involved for a very long time. And in fact, we're really now we can really say that we have completed essentially phase one. So we have literally 4 genomes left for that, and they are already in processing. So basically, we'll consider phase one completed at the end of this month. And this means that we have at least one reference genome representative of each order of vertebrates. But of course, the goal is to then extend in families, gender, and species. Now we have more than 260 genomes because we have generated many more, but we're trying to be able to capture all the sort of diversity of vertebrates at the high level. So having one representative for each order was the logical step. This data is available, so I don't know, it depends on what you do, but this data is available publicly with almost very, very little restrictions. And we also deposit the raw data and all the intermediate files in what we call the GenomeArk. It's our online repository. where you can see all the species, download the data, and so forth. So again, like I said, we are really almost done, -5, actually -4 now. So stay tuned if you're interested in this. The goal after we have completed genomes is to study and reconstruct the phylogenetic vertebrates, study chromosome evolution in vertebrates, comparative genomics of traits and disease, conservation genomics, and so forth. And of course, it's an open door consortium. So if you're interested in contributing to these studies or about their ideas, please reach out. Now, the idea is to generate as complete and accurate as possible genomes. So we use long reads for that. And essentially the way that was described recently to achieve this is this idea of telomere to telomere genome assembly. So basically something that has no gaps inside the sequence, and there are no structural errors, and at the base level the sequence is almost perfectly accurate. However, this is still very manual and very work in progress, so there is no tool suite for that. So we aim to fill this gap by developing tools in the lab that we can use. Again, this idea of C2T genomes is very recent. The first, let's say, large genome, like mammalian-sized genome that was published was, of course, the Human Genome in Science just a couple of years ago. You can see that even just a couple of years ago, there were a lot of gaps, the black bars here in all human chromosomes, and these have been filled. And now we're slowly getting there for a number of other species. So as I said, gfastar, we wanted to have something that could be developed fast, but also be efficient in terms of runtime and so forth. So we decided to stick to C++. And try to not be too complicated in terms of what dependencies you have. So we stick to the standard library. It can process files quickly on the fly. It can generate QC, summary statistics, and it also can manipulate the files and the inputs so that we can, again, more efficiently do what we aim to do. Now, if you're not familiar with C++, is one of the first many compiled programming languages available. It's been out there for a long time. It's very fast compared to, I don't know, Python. It's portable. It's object-oriented programming language. It has a rich library. That's why we choose it. So this is an outline of the library and the tool suite. Basically, as you can see, these are individual tools, and we try to have tools for that can perform a variety of tasks in a certain domain. These are the people that have been working on these tools over the last couple of years. Some people from the lab, Gene Myers, who is a very well-renowned computational biologist, the developer of BLAST, for instance, has been helping a lot. So we have tools to evaluate reads, tools to evaluate assemblies, tools to evaluate alignments of the reads to the assembly, tools that are k-mer-based, and tools to do annotation, like teloscope. This is what Jack is going to talk about in a second. All of this is powered by a library called gfalibs that provides input, output, parallelism, file formats, support, and so forth. And the goal is to continue developing tools. So we have a number of them in progress that are, let's say, less stable. So for instance, K-count is a general k-mer counter. This is to evaluate high-C reads. This is to find patterns in the genome. So just give you the general idea. We're primarily interested in assembly graphs, and GFA is a popular format for that. That names, like I said, the tool suite. So the GFA file format, if you're not familiar with it, it's made of essentially a number of subcomponents. So you have nodes that represent actual sequences, in this case, like A, B, and C. Then you have edges between the nodes that connect the nodes. And these edges can represent either the actual assembly graphs, so the overlaps between the sequences that have been observed and inferred from the sequencing reads, or it could also represent gaps in the sequence, but we're stitching them together because we think they belong to the same chromosome. For instance, because we have complementary information from, say, high-C data, okay? So the GFA format can represent all of this information into a single file. And for this reason, it is much more effective, let's say, than FASTA files, for instance, because it contains, it's information rich. There is no information lost during the assembly process. And so the idea for GFA libs and all this tool suite is that you can, of course, analyze this kind of files, but you can also manipulate them, right? So for instance, you can say, oh, I think these connections, these edges do not exist. So basically I'm removing them. And of course I get a different assembly, and you can do this systematically. Now, again, the GFA file looks like this. We'll look at this, yeah, if you're not familiar with it, in the practical. Basically, again, it can be used to capture the product of an assembly, variation between genomes, and a few other things. Like any other file format, it has its own specification. So we have, for instance, you start always with a header line that basically tells the version. Then you have segment lines, which in our previous representation represents the nodes, so the actual sequence, and you can see the sequence next to it. These are identifiers to distinguish the different sequences, and they're called, again, segments. Then you have link lines, which represent the edges. And for instance, in this case, this is telling us that segment 11 is connected in the forward orientation to segment 12, which has to be flipped, okay? So, and there is an overlap of 4 bases between them, which if you look carefully, you can see that now it's CCTT. So if you flip 12, you get TTCC, right? So it has to be reverse complement, and there is an overlap of 4. And the same goes for all the other links in the file. And then you have paths. So I actually forgot to mention that the path here is the gray line that connects nodes through edges. Of course, you can have a complicated graph, but in order to reconstruct individual chromosomes, you have to spell out a path in the graph that tells us exactly a linear sequence on how to traverse the graph. So the P lines achieve exactly that. And you get an identifier for the path, could be the chromosome name, and then a list of the segments. And the edges between them are usually inferred, okay? So in this case, now if we look at this path 14, we can see that basically you can reconstruct the actual sequence by taking 11 plus, then there is an overlap of 4 with 12 minus, which is essentially this link here. So we only add basically another letter here. And then you have 12 with 13. And essentially you can see the reconstruction here, how it gets spelled out. Okay, so GFA stats was the first tool in tool suite, and basically can read this kind of file and do manipulations. Okay, so you will have assemblies as an input. You could also take a FASTA file as an input and then convert it to a GFA and vice versa. Of course, there is more information loss if you have if you go from GFA to FASTA file, then vice versa. And then it will represent the sequence internally in terms of these components like segments, gaps, edges, and paths. And then we can create the graph. We can manipulate the graph with a Swiss Army knife. We can generate statistics, or we can generate new assemblies out of that. The statistics, they are, some are more canonical statistics like the N50, which if you have done anything that relates to assembly, you're familiar with. Some are a bit more exotic, let's say, like the area under the curve, which is an alternative to the N50. We've benchmarked this with other tools, and essentially we've tried to be very comprehensive so that also all the statistics are there. That's what the blue column here represents for GFA stats compared to other tools that have a similar goal. This is how it looks like, an example. So imagine you have this input with 5 sequences, some gaps in them, and so forth. You get all these numbers right away. It's parallelized, so it's pretty quick. There is a GFA2 file format, which we can also handle, but it's not very popular, so I'm not going. It's very similar, but it's just a different way of specifying things. But it didn't really take off, so it's supported. So in case you have some applications. Then we have the Swiss Army knife. The Swiss Army knife, again, it contains a lot of like a Swiss Army knife. Literally contains a lot of tools and utilities to do manipulation. For instance, we use it to project onto the graph information from scaffolding technology. This is an example here where basically you take a GFA file, provide a golden path file, which is a typical file format for scaffolding, and you get a GFA output it's still in the same coordinate space of the GFA, but has integrated additional information from scaffolding. Because typically what you do instead is that you take, let's say, a FASTA file, apply some transformations in the, let's say with the AGP file, we get another FASTA file, but then it's impossible to relate the two. Whereas in this case, you're only adding information, but not really changing anything. So we use this, as you can see, to generate a GFA plus AGP file that does contain the same information as the FASTA file. This makes the whole process more transparent and reproducible. And for that reason, we basically integrate in our assembly pipeline GFA stats all the steps, either because it's just doing the QC or because we use it to manipulate actively the sequences. Now, that's one thing. We, for instance, support by this approach. We've been working with Anton Korobeinikov, the developer of BandNG, which is a popular tool to visualize graphs. You can now see the gaps in the graph thanks to this combination of tools. And therefore, you get the reconstruction of all the chromosomes and where the gaps are. Like I said, it's a pretty robust tool. So we tested it on the mistletoe, which is a 100 giga genome, and it runs in 30 seconds on a 32 core machine. So it's pretty efficient. And of course, it runs in 1 second on a regular genome, so you don't have to wait to get the results. And then, this was one of the first tools, but then we started to expand. So we added tools first for row reads, for alignment. So I'm gonna just go quickly on talk about these two tools. So RDeval and GFA align. So, again, you could get typical outputs like number of reads, coverage, largest read length, and so forth for RDeval. And the alignments will give you information about the number of alignments to a graph. In this case, we'll have about alignments to a graph of sequencing reads, orientation, average coverage, and so forth. And you can also do some manipulation. So for instance, one manipulation you can achieve with this, again, we're combining this with the visualization from BandNG. So we can add the edge support to a graph. So we take a graph, an assembly graph. We overlay on top of that the alignment of the row reads, and then we call support to the edges in the graph by basically making, in this case, not the edges sticker if they have more reads that support that particular connection. Okay, so again, this is useful if you want to evaluate how consistent the graph is with the raw data. Then I mentioned we have a tool for doing K-mer QC. It's called Creek. It's essentially building a De Bruijn graph from the reads. I want to go to the practical as soon as possible, so I'm going to skip over a little bit, but I'm sure you're familiar with the concept of K-mer. So you take a subset of sequences of a certain fixed length, and basically by doing so, you get information about a particular sequence. So in the De Bruijn graph context, this means if you take, let's say, a read, you chop it into all the sub sequences of a certain length. Like in this case here, from this read, we get GAC, ACC, and so forth. And then we can build a De Bruijn graph, which literally spells out as edges the overlap between the different k-mers. Okay. And then, well, this is typically stored in a table, like the one you can see here. And then we can use this table to assess the quality of the genome. Okay, so as we add the reads, the tables becomes more complex because it contains more information about the graph topology. And then we can use this information to evaluate the genome. Okay, so again, this has been implemented as a in C++. We use hashing, 2-bit encoding, disk and memory usage are kept to a minimum. It's fully parallelized. That's because you typically working with k-mers makes everything a bit more expensive computationally. And the goal we have is to do consensus accuracy estimation, error correction, and subgraphing of the graph. So for consensus accuracy estimation, basically we have improved on an existing method that, again, if you're doing some assembly, you're probably familiar with the QV estimation using Mercury. The problem with Mercury, one problem with Mercury is that there is essentially a bias. So we are always overestimating the QV. And here in this case, we capture more missing k-mers that are a reflection of errors in the genome. And therefore, we can be a little bit more precise. This is an example of the output from the tool. And then we can also generate some fancy outputs that I'm not going to describe in detail, but basically we can, for each base, measure as many information as possible about the k-mers in the De Bruijn graph for that base. So this is what we're seeing here. So we have every row here corresponds to a position in the genome, and we're measuring the k-mer counts in the De Bruijn graph. And you can see essentially even the pattern suggests that there is an error in this position. So we're trying to basically use this to really pin down where the errors are, which is another issue with Mercury, which doesn't give you the position of the errors, but only the estimate of the QV. I talk about subsetting. So this is an example of subsetting the graph. Literally means projecting the raw data information on the genome assembly. Okay, so here is just an arm of a chromosome, and this has been generated with Creek and visualized with Bandage NG again. So basically, let me zoom in a little bit. The blue line is our reference genome, the one we reconstructed. And then here we are adding the complexity of all the information that is present in the raw reads, but didn't make it into the assembly. So what we can see is the gray ones here, they basically spell out alternative paths that exist in the raw reads, but that are not in our reconstruction. Now, this could be just a sequencing errors. In many cases they are, but in other cases they may actually suggest alternative paths that would fix errors in the assembly. So for instance, this is a more obvious one. So the red points here, they correspond to k-mers that are never seen in the reads, but are seen in the assembly. And if you can see, there is an alternative path here, which suggests that probably this is the actual real sequence for that particular position. So again, the goal is to even visually, but also programmatically identify and potentially correct these errors. This is a bit more zoomed in view. You can see, I find it kind of fascinating that you can see this web of potential alternative paths in the graph that are made visible with this tool. And there we go again. Oh, here we have two errors. In one case, you can see there is an alternative path that would potentially fix the error. Here, it looks like we don't have an alternative path in the read in here either. Okay. We have also programmatically implemented a variant calling feature. This is more of an alpha version of it, but the idea is that we can now systematically call these variants in the genome and provide them as a VCF file. So you can then load the VCF file in IGV if you're familiar with that tool. Okay, so now I'll pass it on to Jack. He's going to talk about teloscope, which aims at telomere annotation. Maybe I can keep the slides.
Please,
okay,
because there are only few. Yeah, so telomeres are very interesting. We know that they have a protective role in the cell. We essentially have telomeres in basically all of the chromosomes, but there are also telomeric regions outside of the ends. They're called interstitial telomeres. So we saw the potential of exploring these regions not only because of the medical applications, but also because the telomere length is being associated with several traits, longevity, etc., etc. And also, having telomeres is also a sign of a good assembly, right? So if you have a very fragmented assembly, it's very unlikely that we'll have telomeres, especially if you have like low coverage at the ends. So, do you mind, Giulio? Next slide, please. Okay. So the way telomeres have been studied in the past decade was essentially just exploring this canonical repeat motif, TTA-GGG, which is the canonical motif in vertebrates. Depending on the group, you can have a different motif. Like in plants is different, in C. elegans is different. But also in the 80s and 90s, there were other repeats that were identified in humans. We have TCA-GGG, TGA-GGG, and TTG-GGG, which is just a fraction of the pool of potential telomeric repeats. And there are some articles that have been exploring the diversity of telomeres, and they found that essentially most of them are just neglected. No one really studies them. Okay, so next, please. Okay, also, there is a general lack of consensus in terms of we know what a telomere is, we know where they are, but the boundaries are not very well defined. So the modern approach to characterize telomere regions is to assume or maybe explore if there is a transitional region called the telomere variant region that is reaching non-canonical repeats, and the telomere or telomere that people know is basically reaching the canonical repeat, but we don't know if we have a single variant region, we have multiple variant regions, and essentially every time we have a new variant region, we are essentially creating two new boundaries. So the problem is a bit complicated, but once you establish some clear rules, you can annotate these regions. So next slide, please. Okay, so that's why we decided to implement teloscope to have a fast, scalable, useful tool that is able to characterize telomeres. And the way it works is, Giulio?
I was going to just add that, of course, we are developing a tool suite for telomere to telomere assembly. So we need a telo
Oh, yeah, definitely.
tool in the tool suite.
Yeah, so what teloscope does is it takes an assembly as an input in either FASTA comprised a GC FASTA. So we explore different paths. Julia was talking about paths in parallel, and each path is analyzed, Julio. So we decompose each path by its segments and gaps, which are just the nodes and the edges. And every node or segment is then analyzed using sliding windows. So in each window, we look for the telomeric repeats. To do that, we use this data structure called prefix trees that essentially allows you to look for several patterns in basically linear time. The space complexity is a bit high, but still it's more efficient than looking for the pattern, the repeats individually. Go ahead, Julia, please. So once we have the coordinates of where these matches are, we can then also compute window metrics like GC content, entropy, channel entropy. We know how many matches there are. We know the density of these matches. And with these intermediate outputs, we can generate the final annotation in bed and bed files. Giulio, please. Yes. So far we have the matches, and we essentially need to connect the matches. So we have some parameters. I'm gonna talk about this later, but essentially, if there is a cluster of matches that are very like close together, we can assume that this is a telomere block, and then we infer the coordinates from these blocks. So just a few examples of what what is the advantage of teloscope over other tools. I'm gonna show you this example. You please. Yeah, so this is in the Gorilla. We're gonna use the Gorilla assembly in the practical session. And essentially we have the chromosome 10, next Giulio, please. The paternal chromosome 10, and we see that 60K, which is another telomeric annotation tool, basically misses 3 kb just by not including the non-canonical repeats, which, I mean, it's fair. The annotation is very nice. 60K also does that, like the look for canonical repeats. But if you're interested in capturing the whole telomere, then telescope is a good alternative to do so. And if we think about it, then this particular chromosome is pretty much like this model. Julio, next, please. Where the TBR is basically a mosaic of canonical and non-canonical repeats, and we only have one. Primates are usually like this. There are some chromosomes that don't show a variant region. And finally, because you know where the boundaries are, you can measure the telomere length. Giulio, next, please. Yeah, so if we compare the annotation of 60K versus telescope, basically telescope is able to capture either the same, annotation is pretty much the same, or we capture a longer telomeres just by capturing the variant region as well. So we find this to be very useful. And that is essentially what telescope does. We just released the first version, so you'll probably find new versions in the upcoming months.
Okay, thank you, Jack. Yes, and then we're gonna test it in a minute. So, we designed this initially for our internal purposes, then we felt that it was useful for others too. And so we are really encouraging users, basically, and you guys in this case, to ask for new features, new algorithms. We're also trying to integrate additional features and algorithms on our own, of course. We're also trying to make the tools more efficient in general. But again, feel free to open GitHub issues, and we'll definitely follow up on that as soon as possible. Of course, you can find this. All of the code is available in GitHub, and there are releases, and that's what we're going to use right now. Okay, with that, I think I can stop sharing. Maybe I don't know if there are any questions, comments, curiosities, or if not, we can just move straight into the practical. I don't know how you typically do this, Damon, but
I mean, yeah, questions here is fine. I was going to ask, so this is effectively going to become a one-stop shop for everything you need during an assembly.
that's the hope, yes.
Nice, very nice.
Trying to make everything, also trying to make everything compatible without too much attic, that sort of thing, yes. And again, for our internal purposes, we're using a lot of these tools already, and they, again, communicate with each other a bit more easily. I mean, we're not like trying to, take over everything, but at least what we can do, we'll try to. Okay.
Okay, cool.
All right, so I think I should share my screen again. Let's see if we can make this work. So basically, the way it would work, you take the GitHub link to the gfastar 2024 session. And at least that's how I do it. I don't know if there are better ways of doing this. And then you go to Gitpod, workspaces, you can create a new workspace. Well, let me do that so that we do exactly the same thing. So if I want to create a new workspace, where is the button? Huh?
Go back onto the repo.
Sorry, what?
Go onto the GitHub repo.
Here.
Yeah, scroll down.
Open Gitpod. Okay. That's make even easier. All right, so you don't have to change the settings. 4 cores, 8 GB of RAM, and storage are perfectly fine. So we'll initialize the repo. We have some test files, and the binary files for each tool will be already available in the repo. We have releases with binaries for Mac, for Linux, Windows. So, and honestly, it would compile if you just make into the folder, would compile most of the time, because we're trying to keep it simple, not having millions of dependencies that need to be there for this to work. But again, if you have problem with compiling, let us know. Okay, so I'm gonna wait until the inputs are downloaded. In the meantime, you can see here on the left, we have a number of things that have been pre-fetched in the repo. So we have some FASTQ files. These are the binaries, okay? So gfastats, Creek, RDeval, teloscope. These are the 4 tools of the tool suite that we're going to showcase today. The Gorilla is taking a little longer, but the moment we get that, we can get started. And I'm going to keep it simple. So basically, honestly, you could even stop listening to me and do it yourself, because it's pretty self-explanatory. But basically, we have MD files for the 4 tools. So README files for the 4 tools in the same folder. We're just going to follow step by step. The only addition would be that I can give a bit more of an explanation, and Jack can also give a bit more of an explanation. So the Gorilla is, where is it coming from? It's not from Amazon, but it must be very slow. Well, I guess it's probably Gitpod that is not particularly fast. But
I mean, normally it's not too bad. It might just be because everyone's doing it at the same time.
Yeah, because mine is loaded already.
Oh, really? Okay.
Yeah.
So maybe, and the way it works in Gitpod is that once it's prefetched once, I think it's always available, right?
That's how it was last year. They've changed a bunch of things, so that's no longer the case. So every time you load a new pod, it's a fresh pull from everything.
I see. So I shouldn't have done what I did before. That I was deleting stuff.
I should have changed the assembly to something smaller then.
Okay, why don't you, Jack? Why don't you take over and start with teloscope then? So that, because you already have it, that I will take over.
Oh, okay.
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 We'll have to wait. I have to stop sharing. Here we go.
Yeah.
So we start from teloscope and then come back to the other tools.
Mm-hmm. Okay. Can you see my screen?
Yes.
Okay. Perfect. All right.
Maybe
So
if you can like comment plus or something, you can make the whole thing bigger. No, but
okay.
literally the size of, yeah, exactly.
Okay. All right. So let's start with the help, just to have an idea of what the software needs or does. So F is just file. You have the input file there. We have FASTA files. All of them are zip, but that there's no problem with that. You can read it directly. And with O, we are just specifying the route. There's also this teloscope MD you can access. I'm gonna share it in the chat, so it's available for everyone. Where is the chat? Okay, I don't see the chat. Okay, there we go. Okay, so let's go back here. So the first step is to establish what is canonical. And this flag exists because we don't want this tool to be specific for vertebrates, but the idea is to just analyze whatever you want. Like we have a C. elegans assembly here. The canonical pattern in C. elegans is TTAGGC. So what the tool will do is just essentially set TTAGGC and its complement as canonicals. And then with P, the other patterns that we want to explore, you can use the UPAC nomenclature and essentially use N and NGGG if you want to explore 64 different repeats. I would I wouldn't recommend that because 64 is a lot, and maybe not as informative as just exploring the top 10, top 5. Okay, so W window, just establish the window size. Because we are measuring GC channel entropy, the longer the window, the smaller the number of windows you will get. So there's a trade-off. If you're interested in exploring particular changes in windows, maybe that's for you. If you don't want it, that's also okay. You can just use the default. The step is just how much, uh, how separate are each window. So there's a 500 overlap by default. This is multi-threaded, so Gitbot allows us to use up to 4 threads, so we can set J to 4. Or if you don't use J at all, then by default you will use all the threads available. Okay, so we're gonna play with L and D. What is L and what is D? So you have all the matches, the coordinates of all the matches in the assembly, but with D, you have a distance allow that will help you merge the telomere blocks into longer blocks if you're interested in that. So if you use a very small D, you're probably going to get very fragmented telomere blocks. But if you use a super long D, you are probably gonna get a lot of noise, like two true telomere blocks and something random in the middle. So I'm using 50 because it's not that large, and we can play around that. And with L, we essentially just decide what is the cutoff to generate the output. 500 is fair. If you're interested in only the telomeres, you can use like 2K, 1K. But if you're interested in the interstitial telomeres, then you can lower this L to something like 200, 100. You're gonna get a lot of hits. So there's always a trade-off. The other important parameter is probably K, which is just to keep the window data. If you're not interested in the entropy or the GC content or the densities of the windows, that's okay. You don't call K, and the software will use a lot less memory. So let's start with C elegans. So here we're using this genome assembly that is already in Gitbot. I'm using 4 threads, window of 200 nucleotides, step of 100. It's canonical CTA Because I don't want to type a lot of different repeats, I will use TKWGGC. K represent multiple nucleotides, the same for W. And I will use a short distance and a short length cutoff just to see what does it looks like. So let's start with, oh, God. Oh, okay, I get it. We have to add the GC here. There we go. And so what is happening here? We define the canonical pattern. We explore where the other repeats are very interesting. And then in this verbose mode, we're basically iterating over all of these segments, and that's all. It took a few seconds because this is a small assembly, right? So you can print the summary report because we allowed K, which is keep the window data. We can have this nice summary report that will tell you, oh, you know what, how many. This is the number of matches found for each pattern. Some statistics about the entropy, the GC content, etc., etc. Okay, I don't know if everyone is following me up until now. If not, please let me know. Otherwise, we
I
can
finally
continue.
downloaded the Gorilla.
Oh no.
That's okay. But I mean,
Oh yeah. Next time I will probably just use one. Okay. All right. So we have we can explore how these C elegans outputs look like. K is very well, give you the information of all the repeats, but we're interested in these 3 files, telomere blocks. So in this case, the telomere in C elegans goes from 0 to 774, but because we used a small V value, it was fragmented because from 432 to 572, there are almost 150. So we can rerun this, like literally, just I can delete this, doesn't matter. I can change this to 150 or 200 if I want. Gonna do the same, it's gonna take a few seconds. And now we're done. And if we check that again, now we have our full telomere. So we see that there's also plenty of other hits, and that is, some of them will probably be true interstitial telomeres, but we know that for sure the first one is our telomere because it starts from 0. C elegans telomeres are very short. And the last one is going to be our Q telomere, right? So you can decide, you can manipulate this bed file and decide, oh, you know what? I'm only interested in the first and last annotation, so I don't want this. And then you have the telomeres for each, right? Okay. If you don't want to explore the blocks made of both canonical and non-canonicals, if you're only interested in canonicals, you explore this one in this case because the block is made out of canonical repeats. The coordinates are essentially the same. We're gonna explore the gorilla and the bonobo, and we're gonna check the differences. Okay, if you're interested in one particular repeat, you can always explore the counts. And this is the windows where they are, how many of them are. Yeah, usually just a few. If we explore the canonical ones, TTIGGC, TTIGGC, so the complement will be GCCTAA, which is here, and we can see that we have several counts per window. This window is small. And density is just the counts multiplied by the length of the repeat divided by the window size. This means that from 0 to 200, 99% is covered by this canonical GCCTAA, and so on. If you want the matches because you're interested in the coordinates, you want to not troubleshoot this in IGB, you can use this bed file instead. If you're interested in the GC content, well, this is large, not going to open it. It's the same, it's like we have the bed fraps and so on. So C elegans, pretty quick. Now let's go to the gorilla. So we start with, it's going to create this gorilla. So here I'm using different patterns, a larger window, a larger step, a length, a cutoff length of 1,000, and a maximum distance between blocks of 50. So this is going to take a few minutes. And maybe less than a minute. It depends. I don't know how much memory we have in Gitbot. But in this case, we don't have the K flag allowed, which means that we are not computing the GC entropy, whatever. So we are only interested in the telomere blocks. And these telomere blocks will appear anytime. Yeah, so if you're not sure what to use, you can always explore different values of T and L. I'm working on an extra module that uses a segmentation algorithm or classification algorithm that will do this for you. But in the meantime, you will find that the telomere blocks are pretty much useful. You pretty much get to the final or close to the final result. Okay, let's just wait. And please let me know if you have questions, comments.
So your tool looks like it would be really good in speeding up a lot of telo finding workloads. Is
Yes.
there any plans to extend it so that it finds it without a seed sequence?
Oh, yes. I didn't mention this in the presentation, but this is just the first phase. In our upcoming plans, we want to also implement de novo annotation, and to do that, we're going to explore k-mers, and we'll see if there is like a cyclic k-mer. So it's like, you have a k-mer that is made out of canonical patterns. You're probably gonna look for loops, and these loops are gonna be very frequent. But if we explore the Brilliant graph, we can also explore non-canonical patterns because we're gonna see branches. We can use those patterns found by the k-mers as input of teloscope. So there's always a trade-off. I was at the beginning of this project, I was torn between using exact string matching algorithms or maybe something that can give you maybe approximate matching, right? The difference is that exact string matching can be performed in linear time, like both in complexity, runtime complexity and space complexity. So it's just a matter of this deciding what the correct implementation is. And for this particular case, the prefix tree will be improved to or replaced by a Aho-Kurasik algorithm, which is just a prefix tree with extra steps. You just allow like different branches to connect. And that is very efficient, especially if you're exploring multiple patterns, right? Yep.
Are you sure it's going to complete, or it looks like
Yeah, it should be. I was
it's hanging?
testing this in my computer.
Hmm.
And Gitpod, I don't know. I should
It
have
should
used
be the
the
same. So I
verbose.
think there is an issue. I'll let you debug the issue. In the meantime, if you don't mind, I can take over.
Oh,
And
sure. Maybe it's just taking some time.
that's also possible for sure. All right, so okay, so GFA stats, the first tool we're going to look at. First, just some housekeeping. So we're basically taking some of the test file that are very small and easy to use. Let me make this bigger as well. And I'm going to move the file. So again, you can copy paste directly from here. Let me put this in the chat. Really just copy paste. There's no point typing these days. So we just move the files that we use for testing, like any other tool, and we have help. It's fairly involved. It's not like a million functions, but there are a number of things you can do. So let's just take a quick look at the. Just to make this a little bit small. Just we're testing right now with I don't know if you've ever done some tool development. The smallest file you can create that has kind of all the features of the larger files is a very convenient test file. So this is what it is. Now you have new lines, you have uppercase, lowercase letters, you have gaps of certain types, other types. You have gaps at the beginning, gaps at the end. So that's what we're just to familiarize. So the logic is straightforward. You just give it as an input, and you get this nice summary statistics out of it. Again, it tries to be the most comprehensive, base composition, gaps, segments, masked bases, paths, and so forth. Then you can do some. I'm gonna skim through some of these, right? You can create a tabular output. You can change the locale if I don't know you're in Russia. You want to change the characters. You can do that. You can create outputs that are per sequence. Okay, so for instance, instead of sorry, you can create this output here that gives you, for instance, N50 values, or sorry, NX values for every single potential value, 10, 20, 30. This is useful for plotting purposes. And then you can also create sequence report. That's the next step. So basically, okay, this is not good. It's because it's now called the segment report. Sorry, maybe not. How is this called? I did not update the help, unfortunately. Should still be called set report. Okay, so don't use this function. It was working in the previous version, but it's good that we are testing together. This would have given you per sequence output, so I'll make a note that I have to fix that. good thing about this is that you can convert between file formats. So I was explained before. Of course, we can imagine to output the same file format. We're just stripping the new lines. That's not particularly useful, so we don't keep those, but we keep everything else. But you can change, the way it looks like. So we can, for instance, put them on, fix it the new line lengths like this. We can get subsets of the input. So for instance, here, we're only asking for one sequence in particular and generate the output. We can also, this can be a bit more sophisticated, so you can provide a list of sequences. In this case, I'm providing only one, but this could be a list of sequences that you want to subset. You can also use specific coordinates. So for instance, if we try this out, here I had predefined a bed file that contained a set of coordinates that we were interested in subsetting. As you can see, our original input gets stripped. The E stands for exclude, so we're basically excluding portions. So like for instance, here we're saying exclude the header 1 from 0 to 5, which means completely removing the sequence. In other cases, we're only keeping here one nucleotide because we're keeping we're discarding from 0 to 3 and then from 4 to 5. So we basically keep 3. So we can likewise do the opposite operation. So we can include only those sequences. And so we get only the sequence that we want to keep from the bed file. You can get the size of all the components very quickly. So, and when you say components here, it's like the scaffold sizes. So here are the size of the sequences, the contig sizes. So basically, when you have a gap, you split. This would be the segments in the GFA notation. So for instance, header 2 had a gap in between. So you can see that there were 2 contigs or segments, one of 3 bases, the other one of 1 base. And you can then, of course, use this for plotting purposes as well. And you can get also the size of the gaps pretty quickly. We can also get this AGP golden path output. I may not be familiar with how it works, but this is a more comprehensive overview of the whole thing, including both segments and gaps in the same file. I would say the AGP is something that looks like the GFA file in many ways, but the AGP has been there for 30 years. So we can output this also as bed coordinates if for any reason this is more useful. So instead of this, we say, let's say we want the contig coordinates in bed format, and now we get them like this. So for instance, this is very useful when you want to quickly show where the gaps are in a genome. So you would get the coordinates of the gaps, and then you can use this in IGV or in other software that accept the bed files. Then we can do sorting. Okay, so we can sort by size. We can sort providing a custom, we could like largest, descending in terms of the name. We can give it a custom sort list. Okay, so in this case, I had predefined this random 1 sort list. It happens to have a header 2 before header 1, and we get this also in our output. Then we can convert. Sorry, this was the FASTA file. We can look at GFA formats as well. So GFA2. Again, here we're taking an input that is GFA2 and outputting another GFA2. Not particularly useful, but of course we can also do the conversion from GFA2 or GFA1 to FASTA file. Okay, so this is the same file that I've shown you a second ago, which looks fairly complicated here, but this equates this when projected into the FASTA space. Of course, we can convert between GFA1 and GFA2 and vice versa. Okay, so there we go. You see that it's very similar, but there are some subtle differences, like link lines are called jump lines and a few other things. We can, well, I'm going to skip all this kind of conversions, but they're pretty handy. You can also look at files that have no sequence in them. This is fairly common when you use Bandage because loading the sequence is pretty heavy, so you just skip that. So in this case, the sequence in the GFA file is replaced by asterisks, by stars, and of course, if you ask for a FASTA out of this, it doesn't make much sense, but you can still do that. You can homopolymer compress. Homopolymer compress basically means that if you have runs of T's or runs of C's, they all become a single letter. There are a number of applications in the T2T space for that. So here, you may not see that it's a bit subtle, but if I do not homopolymer compress and compare, for instance, here there are 2 A's and here there is only 1. So that's what this is about. We can find the terminal overlaps. This can be pretty handy. For instance, if you're looking for a circular chromosome or something like that. So let's see. So here, you have this GFA file, and it's a bit convoluted, but if you try to discover terminal overlaps that are perfect overlaps, we now start to have more link lines that give us that information. So, of course, we can look at paths as well, and we can also discover paths in the graph. Okay, so take this example. Now I have removed the paths from this particular sequence. Now we're still looking at the original FASTA file. But now if I, so now if I take this file where I strip the path lines for testing purposes, there is no more paths in there. And now I use the discover path option. I can find them again. Okay, and I can output this as GFA or GFA2. And you can see now the paths are back because they are implicit in the graph. So that's also handy sometimes. We can, that's what I was showing you in the slides, or we can superimpose an AGP file. Okay, so imagine we have an AGP file that suggests we have to connect some things. Like in this case, we're connecting header 1 with header 2 and so forth. And so basically by combining the FASTA file with the AGP file, we get a completely different sequence essentially, or a sequence that is a linear combination of the original sequences. And then we have this Swiss Army knife, which has a lot of instructions. So they're detailed. There is a README file in the GitHub with all the instructions that are available. I'm going to show some of them. Okay, so typically there is this SAK file, which contains the instructions. Okay, so for instance, this is a very simple one. We're just saying we want to reverse complement header 4 and then reverse complement header 3. So this is a sequential set of operations that we perform. Okay, so if I check my test file and now I use the Swiss Army knife to manipulate it, if you look at sequence, what was that? 4 and 3, they should be reverse complemented, right? So TGA has become ACT and et cetera, okay? And the same goes for header 4. So that's just one manipulation. If we take these other example files in the folder, we have join instructions, split instructions, excise instructions, invert, remove, resize, and so forth. Okay, so let's just, so what I'm doing here is basically I'm taking all these sequential instructions one at a time so that you can see how this performs. Okay, so we start with this file. We do one manipulation here, which was join header 1 and header 2. And now you can see, and we call this new component scaffold 1. Now you can see we have now scaffold 1, which is a combination of header 1 and header 2 with a gap of 5 bases in between. So we can do this kind of manipulation, okay? I'm not gonna demonstrate all of these. Of course, they will also work, you can generate GFA outputs, but just to keep it simple, again, this was the original file. Let's say I take 4 instructions to the Swiss Army knife. The sequence is highly manipulated. And these are all pretty efficient operations, pretty inexpensive. Okay, so this concludes GFA stats. I don't know if you have comments, questions, or if you were able to follow. We have 15 minutes left, so I'll just, in the interest of time, I'll just move on to the next tool, Creek. So again, works in a similar way. Now, there is this validate function that's what we use, okay? So basically, again, let's take some test files first. Okay, so we just run this to take the test file specific for Creek. Now, this was supposed to be 2024. So we move the files. So the way this works is that essentially you provide a assembly as a FASTA file or a GFA file as an input and a set of reads to use to evaluate. Remember, Creek is using k-mers to evaluate the assembly. So if you run this, the first thing that happens is that we create a k-mer database. Of course, again, this is a very small example. And then we generate these QV scores that in a very simplistic case, like this very small example, they don't differ between Mercury and Creek, but in Creek, again, we have less false negatives in terms of missing k-mers. It's a little bit more accurate. You can run this with multiple FASTQs as inputs or gzip FASTQs. Should this be one? Okay. We can generate a database and then reuse it. So that's pretty handy. So if I now run this and I ask for a Creek output as an extension for the file, I get only the database. You can see it's at the Bruin graph because it says there are a total number of edges here, right? So it's a slightly different format from a k-mer counter, for instance. And then we can reload the database. Now, again, this is a very small, random one. It's good just doing the same thing, but it's going to be extremely more efficient if you use the same database over and over. It will take 1 second or 10 seconds, even for large databases. And you can combine, you can use instructions like union to create multiple databases individually and then combine it into one, and then use that one to do the validation. Okay, so yeah, just to get a sense of what these things looks like, we can run this on a slightly bigger input. So for instance, we have this, what is it, 20 meg genome. It's just a fake sequence probably. And then we can do exactly, so we can basically generate, in this case, a first database from a read set. Okay, so we generate that. It shouldn't take very long, but of course it's a bit more involved because we're counting all the k-mers. And then let's say we generate another database from another read set, and then we want to combine them into one. So you can see this is the first database. Then we generate a second database, and then we can use a union operation on these two to combine them. That should also be pretty quick. Okay, you can see now they have different stats. The union should be pretty fast. And then we can run quick validate on the filtered _ union quick file, which loads the database pretty quickly, as you can see, and then goes through the 20 million bases to do the estimates of the QV, and also you can output the individual positions that are problematic. So in this case, you get more reasonable QV. Again, it's a very simple example, so there is no difference between the two estimates, but in other case, you would get a difference. So this concludes the quick demo. And then for RDeval, I wanted to show you just a very few things. So basically, RDeval, again, does in principle what gfastar does for assemblies, but on raw reads. Okay, so again, you have your help, and you can run it on FASTA files or on FASTQ files. You would get, the number of reads, the total length, average read length, so statistics of some kind. Or of course, here we're using a FASTA file, so we don't have read quality. That's why it's 0. But if, let's say, we were running this on a FASTQ file, the one we were using before for Creek, we would get, you can see it's pretty fast compared to, for instance, computing the k-mers. We would also get this average read quality. This is a typical read quality from hi-fi data. To be honest, I don't think this is a number, this estimate is correct. It's a separate discussion, but this would be too high. But that's how they do it in the hi-fi FASTQ files. So the other thing that you can do is you can use this for filtering. This is very helpful in the context of T2T genomes. So basically, T2T, one of the main components of a T2T assembly is this ultra long nanopore reads. So if you want to filter, for instance, the reads to get only the ultra long ones, which are defined as above 100 KB, you can do so by using this -F option, and you can give it the number of natural language or mathematical language inputs, and it will, for instance, in this case, will filter for read lengths that are above 10 base pairs. Okay, so if you, of course, you may want to do this filtering and then get the files. In this case, we're only running this and getting the stats, but of course, you may want to output, for instance, all the reads before and after filtering, right? So in this case, we just get the stats of the reads before filtering. Then imagine we want to filter only for the reads that are above 10,000 base pairs. And we also want to create a FASTQ file that contains only those reads. Here we go. And then, the idea is that if you look in our RDeval repo, there is also scripts to plot read quality, read lengths, and other things like that, because essentially what we do here is that we can output a number of metrics for each individual read, and then use this for, let's say, plot them in Python or R or other tools, right? So in this case, for instance, we're asking for the output of a histogram. So here you can see we're looking at a very simple input. So this was the original input. There are 9 sequences of I think 100 base pair long in most cases. Some of them are 98 and some are 99. In fact, there are 2 sequences of 92 base pairs, 5 sequences of 99. So essentially, this is the histogram of the read lengths, and these are the individual read lengths. Okay, so you can plot this. Let me see if I can show this. Yes, so if I run this on the filtered FASTQ, I will be able to see, how the histogram looks like for a more comprehensive. And there are other options for similar kind of outputs that you can then use to play around. Okay, so we're almost at the top of the hour. We, maybe rushed a little bit through the presentation, but all the material is available to you, so it should be relatively fast and easy to check it out. If you have any, again, requests or any comments, we're very welcome. You're very welcome to ask, and we'll try to do our best to integrate them. So thanks for staying, especially to Adam and Alex for the whole session. And thanks, Damon, for hosting it.
Thank you for giving up your time for giving it. And yeah, I'll see everyone in the next session then.
All right. Thank you, Jack, as well.
Thank you all.
Bye-bye.